Matthew. Yes, this was a very exciting work that I conducted at Memorial Sloan Kettering Cancer Center as I was a postdoc, as you mentioned, co-mentored by a cancer scientist who's Steve, and then a splicing expert as it is Omar Abdel Wahab. And aside from these, great mentors, we also had the opportunity to collaborate with people across different fields, experts in mutations in p53, experts in KRAS biology, experts in RNA binding biology, and then, to have a complete story, or what we think we were finally able to kind of dissect out this mechanism. So whenever we're ready, I can get started.
Yeah, sure. I just want to remind everybody that, as Seth said, you can use the Q&A function in Zoom to post questions, and we want this to be more of a regular journal club format. So let's pretend we're all sitting in a room together. If there's any question that comes to your mind as we go, I will be monitoring the questions and relaying them to Luisa. They can be technical questions or more about the biology. So feel free to post these questions and ask the Q&A functions. And from this point on, I think we're ready to go, Luisa.
Okay, thank you, Matthew. So let's get started. So this paper came out in August of this year in Cancer Cell. And basically what we describe is the cooperation mechanism between two of the most common oncoproteins found in tumors that coexist, which is mutations in p53 and mutations in KRAS. And although we have known for these coexistence of these two mutations for many years and across different cancer types, no one had ever dissected out how do they cooperate to cause tumors. And we were just excited and surprised that we landed in altered RNA splicing to be that bridge that connects these two oncoproteins to for the pathogenesis, at least of PDAC or pancreatic cancer. Okay, so I got interested in pancreatic cancer 6 years ago when I was moving from my PhD to my postdoc, and many reasons why I got interested in this tumor. As a cancer biologist, I just was surprised to see that in our war against cancer, we're making progress in the 5-year survival rate for many of the cancer types, or many cancer types. But it's sad that for pancreatic cancer, the line has stayed flat, although we have developed really nice models and we somewhat understand the genetics. But part of it is because it's diagnosed, this disease unfortunately is asymptomatic. We don't screen for it. It's diagnosed once this tumor has spread to other parts of the body. So when it's systemic disease, the first lines of chemotherapy that we have, really, they're not curative. They only extend survival of some patients. Immunotherapy is ineffective. And basically, the targeted therapies that are available out there for actionable mutations, these mutations are rarely found in pancreatic cancer. So there's really a need to understand these tumors better and to come up with different ways to treat these tumors and thinking out of the box from all these different cancer types where we have been more successful at treating them. So a few years ago, at the same time when I was starting at MSK, a few groups across the US and internationally, they reported that the most common form of pancreatic cancer, pancreatic ductal adenocarcinoma, or PDAC, actually comes in different molecular subtypes. And these molecular subtypes are basically identified based on the genes that are being expressed in the tumors. So what they found is that mainly when they correlate gene expression to survival, there is this group of tumors that appears in patients that they have called the squamous or the basal subtype. And if patients harbor these tumors, they have a worse outcome, depicted here in blue. Now, when we look at the heat map of what are those features that genetic and transcriptional features that make these tumors, what the scientists all found is that first, the genes that are upregulated in the squamous subtype are genes that are encoding for RNA splicing proteins. So here you see it's a very confined group of genes. All the rest of the genes that are expressing the other molecular subtypes are silenced in the squamous subtype. Now, the other association that they found with squamous is that it has a high frequency of mutations in p53. These are mainly gain of function mutations on p53, which we're going to cover in a moment. So when I saw this, my first question was, is there any association between alterations in RNA splicing and the p53 status in these tumors? So just to remind everyone, mutations on p53, as I was mentioning earlier, are is probably the most common mutations in tumors, but they come in two different flavors. So as many of you know, p53 is a tumor suppressor protein, but it can have two types of mutations in tumors. You can have missense mutations, and there in pancreatic cancer, there are particularly these 4 ones that I have circled here. And these missense mutations actually have been found to provide an oncogenic function for p53. On the flip side, you have another type of mutations, which are those truncating mutations that lead to the loss of the wild type p53. And these are all depicted here in green. As I mentioned before, the most common mutations that were associated to the most lethal subtype of pancreatic cancer were mainly these mutations, the gain of function mutations. So what we did, we started very simple. We took all the RNA sequencing from the TCGA and the ICGC, and we took their raw RNA sequencing, and we started and divided our tumors into 3 different categories, either if they had wild type p53. By mutant p53, I'm referring just to these 4 mutations here, or null p53, so all the truncating mutations. And we started comparing these tumors between them to see what we can find at the level of alternative splicing. So here are the results. What we found is that in this side, what we have is all the comparisons of all the mutations of p53 compared to tumors that have a wild type p53. And on the x-axis, we have this scale of differentially spliced exons to depict to a certain extent how big are the changes in alternative splicing. As you can see, mainly the mutations, these hotspot gain mutations of p53 are changing alternative splicing compared to those truncating mutations. On the flip side, if we compare that mutants p53 versus those truncating mutations or no p53 tumors, once again, we see that these hotspot mutants are changing the way that the tumors use their exons. And importantly, it was this mutation, the R175H, that we saw that it was changing exons the most. So this were just correlative analysis, but at least it gave us a sense of that there was something here that we should dig through. So we started manipulating the expression of R175 in 3 different systems. The first one is I took patient-derived organoids, so these are primary cultures from patients, and in this case it was a patient who had the R175 mutation and a KRAS mutation, and I used short hairpins RNAs to knock down the expression of this mutant p53. I forgot to say, patients, when they have this mutation in p53, they lose the wild type allele. So they have this loss of heterozygosity. So mainly here, we're just knocking down the mutant form. And after we got this successful knockdown, we did RNA sequencing and alternative splicing analysis, and we derived these scatter plot. And basically what we have here is every red dot that we have in this scatter plot are exons that are promoted by p53. For example, if we focus on this exon, it would be an exon that it's found almost in 80% of the cells that have expression of p53, but it would be lost or it would be present in only about 20% of the cells with knockdown of p53. And then in the blue area, what you have are those exons that are preferentially skipped by p53.
Luisa, I have a question here. So you say on the x-axis the fraction of cells that present with the cassette or without the cassette, and not the quantification of how much the cassette is present in each cell?
Correct. So here we have the percent spliced in, And this is how many times in the sample it was read a particular exon, and it's expressed in a percentage. So as I was mentioning here, this exon, for example, would be present or would be retained in the cells that express p53 in about 75% of the reads.
All right. Thank you.
And then it would be only found in about 10% in those ones that have the knockdown, suggesting that mutant p53 retains these exons. Thank you, Matthew. Okay, so after we did this first model, we did another couple, which are murine models. So we took this cell line that is generated by this genetically engineered mouse model that bears R175 KRAS mutations, and similarly, we just took this approach of knocking down p53, RNA sequencing, splicing analysis, and very similarly, we were seeing some exons being promoted in the presence of mutant p53 and other ones excluded. Lastly, we compared the wild type p53 versus the mutant p53 in a model that is the panning organoid. This is basically a precancer state. We wanted to see how early, when they gain p53 mutations, are the exons being differentially spliced. And so what you can appreciate from all of these models is that there was kind of like this swapping of exons. Then we asked the question, is there anything common about all the promoted exons and all the excluded exons? And it was surprising to us. We found that there is a sequence specificity on these exons. Here on the left, we see that the repressed exons were actually enriched in purines. They were rich for A's and G's. And on the right-hand side, we can see, and on the right-hand side, we can see that the exons promoted by p53 are highly enriched in cytosines, and we call this the poly-C exons. So for the rest of the paper, we decided to focus on the biology of these promoted exons by p53. So when we look,
With a quick question.
Yes.
So about this poly-C motif, do you know in what fraction of p53 altered exons this motif was present?
Yeah, so when we look first at all RNAs, coding and non-coding, and it was present in about 60 to 70% of the exons. So it was somewhat of a robust sequence specificity. Mm-hmm. It became more strong in the coding exons. So after you just filter for coding exons, it was higher than that. Mm-hmm.
Right. Thank you.
Great. So thank you, Matthew. When we looked at where those poly-C tracks are found, as I mentioned, they were found in the axon, but they also extend somewhat to the splicing sites and in part to the intron, suggesting that it's a very conserved sequence, not only within the axon, but extending to the splicing regulating sites. Just to give you a flavor of how we were seeing these changes. These are the sashimi plots, or the raw RNA sequencing reads from the patient-derived organoids where we have R175 or knockdown. And here it's the reads for different exons. I'm just giving you here an example for this mRNA that we found alternatively spliced by p53. It's GAP17. In humans and in mice, there are two isoforms of these GAP17. The only difference is the retention or exclusion of these exon 17 or poly-C exon. So what you can see here by the height of the peak, in the presence of mutant p53, there was more reads for these exons than it was for the knockdown. In the murine PDAC cell lines, the same. You see a high read in the mutant form, but very little, if any, in the knockdown. And lastly, in the murine panning organoids, where we were comparing mutant versus wild type p53. So this was exciting to us. Later, we went and we asked, well, you remember that there were more mutations of p53, these gain-of-function mutations of p53. So we went back to look if other mutants of p53 could actually be promoting also the retention of this poly-C axon. So we took a handful of PDAC organoids, and we did this targeted PCR to look for the retention of this poly-C axon. And what you can appreciate is that tumors with wild type or with null p53 did not retain that poly-C axon, but more so all the mutants, these gain-of-function mutants with a different extent. suggesting that these splicing changes are not only associated to the R175 mutation, but to other gain-of-function mutations. So then the question became, well, p53 is a transcription factor. How could it be that it is changing RNA splicing? So from the RNA sequencing that we had obtained from all these models, we went back and we started looking if any splicing factor, if its expression was being changed as a function of the expression of p53. And we found very interestingly and robustly also across different systems that when mutant p53 was present, there was higher expression of these RNA binding protein hnRNPK. And whenever we knocked down the p53, it would go down, or wherever we did the CRISPR knocking of the mutation, the expression would be increased. So when we look back at hnRNPK to see if this splicing factor could be potentially be regulated by p53, we surprisingly found that indeed the promoter of hnRNPK has different p53 binding sites, suggesting that potentially p53 or mutant p53 could be binding to these sites to promote the expression of hnRNPK. So for that, we did this reporter assay where we measured the expression of hnRNPK when it's tagged to TD tomato in either cells that had the wild type or the normal p53 binding sites versus those cells where we had deleted these binding sites for p53. And we had all these plasmids of expression introduced to cells, isogenic cells with either wild type p53, different mutants of p53, or just null p53. And what you can appreciate here is that indeed the mutants were able to increase the expression of hnRNPK compared to cells that had wild type p53 or empty vector. And as soon as we deleted these CAT-G sites where p53 binds, that expression was significantly decreased, suggesting that the mutant form does have a preference for hnRNPK promoters at this predicted p53 binding sites. Then we did, I mentioned that we had found these poly-C exon inclusion in the presence of mutant p53, and it was similarly to what we found when we did RNA sequencing and splicing analysis in the same cells where we manipulated the expression of hnRNPK, and we found that once again the promoted exons were very much mirroring the effects of the p53, not only for the promoted exons but also for the repressed exons. Yes, Matt?
Yes. What's the overlap between hnRNPK targets and exons that are altered by p53 gain of function?
That's a very good question. You know, we found that it wasn't 100% overlap. If anything, was like 10% of overlap. So there is additional mechanisms that we didn't find or that we haven't found better that definitely is worth looking into. If it's not expressed by hnRNPK.
Right. So I'm not an expert of p53, but my understanding is that this is a multifunctional protein. Is there any evidence that p53 does or does not bind RNA directly?
There is a handful of papers from the 90s and from the early 2000s by sorry, there's a lot of sirens going on. This seems New York City, but it's New Haven now. But anyway, so and it's by the same group. There hasn't been really thorough analysis of p53 binding RNA sequences, but in these handful of papers, they do report that it tends to bind to poly-C sequences. We're actually looking into this ourselves. I'm collaborating with Dan Dominguez at University of North Carolina, who's an expert on RNA binding proteins, to identify if p53, mutant p53 or wild type p53 could bind to RNA directly.
Great, that's interesting, thank you.
Yes, thank you, Matt. That was an excellent question, thank you. So the final experiments that we did for hnRNPK, I was just mentioning Dan Dominguez at UNC. To basically, he had previously published that indeed hnRNPK binds to C-rich mRNAs. And here's just a dot plot of the binding efficiency between hnRNPK and these different sequences in mRNAs. So we contacted Dan and he repeated the same experiment this time around for the poly-C GAP17 exon. And what you can see is that indeed hnRNPK binds very strongly to any just random C sequence and the poly-C exon of GAP17. And lastly, as a visual, we now know that when we knock down hnRNPK, we actually lose that poly-C exon on GAP17. And when we overexpress hnRNPK, we gain again this poly-C exon. So till this part, we had identified that p53 was promoting poly-C exon retention, potentially, or at least some of the events explained by hnRNPK, by p53 directly binding to the promoter of hnRNPK, and then hnRNPK as a poly-C binding protein regulating the retention of these exons. But really, what is the biological consequence of these altered RNA splicing in the setting of a mutant p53? Why would p53 be changing or these preferences in exons? So we took all those mRNAs that were gaining poly-C exons, coding mRNAs, and we asked the question, are they encoding for a particular group of family of proteins, or are they working in a specific pathway? And the results were very interesting. So this is a plot of enrichment analysis. And what we found is that there were several pathways where these mRNAs were potentially impacting, but the majority of these mRNAs were part of a family of proteins called GTPase activating proteins, or GAPs. So what do GAPs do? GAPs help deactivate RAS proteins. So they accelerate the GTP hydrolysis when it's bound to RAS, and then they bring RAS to a GDP bound form, which is the off state. So GAPs are the negative regulators of RAS proteins, including KRAS. And what we were surprised is that it was just not a small group of the GAPs, of the mRNA of GAPs being impacted. Actually, 25% of all the GAPs encoded by our genome were being affected, or their splicing was being affected by p53. So these different GAP isoforms are coding GAP isoforms, and in here you can see in this Western blot that they encode for different proteins. They just have a different size. So whenever they have that mutant p53, they have an extra exon, and that exon, when it gets translated, it encodes for these proline-rich motifs. And at least in GAP17, it's these exon 17, it's at the end of the protein. So basically imagine gaining these proline-rich tails in these GAP proteins. It doesn't interfere with any of the catalytic or membrane binding domains of the GAPs. It's just this additional prolines that are hanging out.
Yeah. How big is the exon itself?
So the different exons in all the GAPs vary. GAP17, the exon is about 230 something. And in here, just to show you kind of a flavor of all the alignment of, these are all GAPs, and these are all the proline domains coded by these poly-C exons. And the majority of them were always at the tail end of the protein.
Do you know if these GAP exons are conserved?
Yes, where at least with the mouse there, GAP17 poly-C exon is fully conserved between mouse and human. We didn't go back farther. Yeah, we only went back to between mouse and human, but at least for this one and for a few others, it's highly conserved. Thank you, Matt. So it was the time to get into biochemistry. So as I mentioned before, GAPs are these proteins that accelerate the hydrolysis of GTP to GDP when it's bound to these RAS proteins. So we wanted to understand if these isoforms of GAP17 had differences in this catalytic activity of GTP hydrolysis. So we first did this in-cell experiment where we overexpress either the poly-CGAP17 or the minus poly-CGAP17. And we did a co-immunoprecipitation for active KRAS. So any KRAS that was bound to GTP, you can pull it down. And then we use an antibody that only recognizes this G12D mutant form to measure how much of the mutant KRAS was still bound to GTP when we had these different isoforms. So the results were very surprising to us. When you have the expression of poly-CGAP17, the isoform that is promoted by mutant p53, the active levels of KRAS remain the same as the control with the empty vector. However, when we overexpress this minus poly-CGAP17, there was a significant drop in the levels of active KRAS. So it was almost like day and night for these two different isoforms of the protein. And also as a downstream effector, KRAS, active KRAS, GTP-bound KRAS phosphorylates ERK, and we can see that this activity of ERK is also significantly diminished when the minus poly-CGAP17 was expressed. So this suggested to us that there was definitely a difference between this capacity to hydrolyze GTP by the two isoforms. So we went ahead and we measured this in a cell-free assay method where we combined either wild-type KRAS or mutant KRAS when they were bound to GTP, and we measure hydrolysis in the presence or in the absence of poly-C or minus poly-CGAP17. And these results were at the beginning very confusing to us. As you can see here, there is no difference between the poly-C and the minus poly-C catalytic activity of GTP. They are identical. They both have that capacity to hydrolyze GTP. But how then could we reconcile these observations in cells where both the poly-C and the minus poly-C had very different effects on the hydrolysis of GTP in the cells? So this said that there was something about the context of the cell that differentiates the activity of these isoforms. So for those of you who are not familiar with GAP biology, so basically GAPs are cytoplasmic proteins, but however, when they need to hydrolyze GTP from KRAS, they bind to the membrane to meet KRAS, which is a membrane-bound protein, to hydrolyze this GTP. So we were curious to know if the poly-C exon addition was impacting the subcellular localization of GAP17 isoforms. And here what you have is immunofluorescence of cells that have mutant p53, or where we knock down mutant p53. And what we stain for cadherin, which is a marker of membrane, GAP17, and the merge, and what we can find is that indeed, whenever the poly-C isoform was present, it doesn't have that capacity to reach the membrane so efficiently. However, when they lose that poly-C exon, now they can gain access to the membrane and hydrolyze that GTP-bound KRAS. So that explains why we were seeing these differences in cells, but not in the tube. So overall, what we had been finding is that indeed this mutant p53, it's promoting the active form of KRAS. And I've shown you through the GTP isoforms, but even when we knock down p53, you can see the significant decrease on KRAS activity. We can also do analysis by signal transduction, experiments in using RNA sequencing from PDACs and from other tumors. And basically now we know that mutant p53 helps to maintain the active form of KRAS, potentially by the splicing phenotype that I've shown you. So as a summary for what I've shown you so far is that we had identified that this mutant p53 promotes the expression of poly-C binding protein hnRNPK, and it particularly promotes the retention of these poly-C exons in gaps that when these guys get translated, they are no longer efficient at reaching the membrane, potentially because they have gained this additional tail of proline-rich domains, maintaining KRAS in the GTP bound form, highly active and promoting tumors. On the flip side, if we knock down the mutant p53, or if they have a wild type p53, they will go through the canonical splicing. They would eliminate this poly-C exon, and they would encode gaps that are proficient at binding the membrane. So yes, Matt.
Right, and I want to point out that in the paper you also show that the lack of RAS inactivation by GAP17 actually leads to tumor growth.
Correct. That's right. So it leads to tumor growth. It leads to maintain KRAS activity and ERK activity. So there is not only a biochemical effect, but also a biological effect that also reflects on the tumor size and on the survival of the mice. And I'm going to start going into that in vivo data.
That's very cool. I want to, before we move on, I really want to maybe bring something of a discussion point in mentioning that this mechanism that you uncovered, where you have an alternative exon that is shared by many members of a protein family, that it in self encodes a whole protein domain or motif, is very rare on its own. The fact that it's also conserved in a mouse is also unusual for alternative exon, and this is what you observe here. And you've shown what the function of these exons are in the context of cancer, but I'm wondering what could possibly be the function of these regulated exons in a more physiological context. And, you know, this is more of a discussion point.
Yes, definitely. You know, that question becomes important not only from the physiological and evolutionary point of view, but also from a therapeutic standpoint. Because if we're now going to start disrupting the retention of these poly-C axon to recuperate or to correct gap activity, we need to know where else is expressed. And are we going to have side effects when we target this poly-C axon? So we did start screening for where, in what tissues, at least from mice at that time, where is this poly-C axon being retained? We found it very strongly retained in ovaries and testes of mice, suggesting that potentially has to do something with the stem cell phenotype or embryonic phenotype. We didn't find it. There was some in the liver, but very little, if anything. But other tissues such as heart, the lungs, stomach, all the GI tract, we didn't find it. Mm-hmm.
All right, thank you.
For sure. Okay, so let's get into the therapeutic standpoint. So yes, you know, dissecting out a mechanism is very interesting. It's exciting. But as I mentioned, we care about the patients. What can we do from this new biology that we learned? So we started thinking about a couple of targeting strategies to disrupt this cooperation mechanism between p53 and KRAS. So the first one was to modulate the spliceosome. So many of you may have heard of this small molecule inhibitor, a splicing modulator called H3B8800. This compound, basically what it does, it prevents the spliceosome from recognizing or binding very tightly to the mRNAs. This splicing inhibitor is currently in phase 1 clinical trials, mainly for leukemias where there is splicing factor mutations. But our hypothesis here was if truly mutant p53 tumors depend on RNA splicing, potentially they might elicit a therapeutic vulnerability to this compound, and if they really depend on the expression of these policy gaps. So we started doing what we call an animal or a mouse clinical trial. So we took mice that have either mutant p53 in red or loss of mutant p53 in blue. And in each one of these groups, we randomized the animals to either receive vehicle treatment or 8800. And here is the survival curve. So mainly all the animals that got 8800 benefited, and their tumors were smaller, and their survival extended. However, the animals that benefited the most were those ones that harbored mutant p53. As you can see here, there was almost a 3-fold increase in the survival probability of these mice when they were treated with this drug, suggesting that mutant p53 sensitizes these tumors to the splicing modulator. When we looked at the RNA-seq from cells treated with incremental doses of these 8800, we found that indeed 8800 decreases the retention of this poly-C exon as a function of the concentration of the drug, kind of like reversing the effects by the mutant p53 at the splicing level. The second strategy that we took to manipulate.
Luisa,
yes?
if you will allow me a quick question. It seems like using an SF3B1 inhibitor is a rather non-specific way to do things here. So in your hands, how many exons or splicing events were altered by the treatment with this drug?
Thousands, Matthew. And it's been reported, you know, by other groups that this drug really, it's, as you said, is very is non-specific. Several mRNAs and their splicing is going to be disrupted. One of these examples could be GAP17, but there were many, many gaps and many others that were not associated with p53. And
So the idea of using SF3B1 in the context of cancer would be in assuming that these cells are already vulnerable to extensive changes in splicing because they are already somewhat deregulated, or they have misregulated splicing, which makes them more susceptible than normal healthy cells.
Correct. So actually the whole concept of using 8800 is that the spliceosome or the splicing pathway can only tolerate one perturbation. Otherwise, it's gone, right? So when the splicing factor mutants, they have already a stress in this pathway. And as soon as you give the drug, that's when they can no longer tolerate it. And that's where you see the vulnerability. In this case for us, is already hnRNPK and p53 are putting a stress on the cells, or on the splicing or the canonical splicing of these cells. And that's when if you give this inhibitor, we were expecting this vulnerability to be exposed. But I agree, this is probably not the cleanest way. This splicing inhibitor targets both wild type and mutant SF3B1.
Well,
so,
it's a case for still many chemo treatments anyway.
Right, and it was also, I guess for us, it was therapy, but it was also proof of concept.
Right.
So then we wanted to do a cleaner way to correct the splicing defect. So we designed an oligo-based therapy. We call it splicing hit oligonucleotide therapy, or SHOT, where we basically use oligos, antisense oligos that can block the splicing sites of these poly-C exons and reverse those and promote the exclusion of these poly-C exons. So in here we designed actually for several shots. These two were the best ones to promote the exclusion of these poly-C exon, as you can appreciate here from this targeted PCR. So we then went ahead after seeing this specific effect on the poly-C exon of GAP17, we went and performed another mouse trial. So we took mice and they all had mutant p53, mutant KRAS. We implanted the tumors until we could see them visible. And then we randomized the animals to either receive a control shot or targeting shots. And after injections or treatments for 12 days, every other day, we found that the tumors that were treated with these shots that were provoking the skipping of these poly-C exons stalled tumor growth over time compared to the controls here. And this was exciting to us because it suggests that manipulating these exon changes specifically could be probably a better way to go after treating these tumors in a cleaner manner. And it was sufficient. We were just surprised to see how just reversing this one effect, it was so potent to decrease tumor size, to induce tumor necrosis, and to extend animal survival as well. So I was going to wrap up, Matt.
Yeah, I think that's a very striking result. I was going to ask another specificity question here. Can you tell us more about the design of these oligos, because you're targeting a rather short, relatively common motif. So how would you go about this?
Yeah, so we definitely, it's tricky, and that's why we test several. So we test for good binding, specificity as much as we can. These are about 25 nucleotides long, high melting temperature. What else? We do have, so they overlap exon and they overlap intron. They overlap the exon by like 5 to 6 nucleotides, and then the rest goes to the intron.
Did you have to use any special kind of chemistry on these oligos?
Yeah, so with the company that we worked, so for the in vitro experiments, we just bought first almost like a lipofectomy type of deal where you just open up the cells and then you just pour the shots in it and then they will go in. For the in vivo experiments, we linked it to this known, I'm blanking on the names, but I think it's lysines. It's a change of lysines that basically it increases the solubility to get it into the tumors. We're now collaborating here with an expert in oligonucleotide delivery. Basically here at Yale, they have a patent really nicely for what's called flipped. So basically it's this kind of biosyringe that you take your shot or your oligo, and it's a peptide that when it reaches highly acidic conditions, it forms an alpha helix, a transmembrane alpha helix, and it delivers the oligo. And that way we can apply it systemically, we can track it, it's fluorescently labeled, and it's also being now used in the clinic. So we're hoping to, further elaborate on this therapy by combining it with this delivery technology.
That's really fancy.
We'll see how it goes. We're just going to start. They have done it with microRNAs and they have great results. And so we're excited to get this started.
Nice.
The other thing that I forgot to mention is this poly-C exon retention by mutant p53 is not specific only for pancreatic cancer, but we also find it in lung adenocarcinomas, in colorectal cancers, and in breast adenocarcinomas, even if they don't have KRAS, suggesting that mutations in p53 activate the RAS-ERK pathway, even in the absence of mutations in the RAS. So basically what I've shown you today is that all these gain-of-function mutants of p53 are associated with specific changes at the RNA splicing levels, particularly of exons that are enriched in C sequences, that this changes in RNA splicing is through the well-known poly-C binding protein hnRNPK, that these changes in isoform do switch the active form of RAS GTPases, and that potentially there is this therapeutic opportunity for KRAS and mutant p53 pancreatic cancers when we manipulate the expression of gap splicing or the spliceosome in general. And with that, I just want to thank, as we were mentioning earlier, my mentors at MSK, but all the wonderful labs across MSK, other universities. We collaborated with Rob Bradley at the Fred Hutch for all the splicing analysis. With the Dana-Farber, we got a lot of their organoids. We collaborated with Channing Durr and Adrian Cox, who are experts in KRAS, mutant KRAS biology, Dan Dominguez, RNA binding biology. And right now, just building my new collaborations here at Yale. And just a shout out to anyone who's looking out for a particular postdoc, if you're interested in my lab. We're a small group, but very vibrant, and we have really nice experiments and projects ongoing with very fancy technology incorporated. So shoot me an email in case you're interested with your CV, and to thank my funding. And that's it. Thank you.
Great presentation, beautiful work. Thank you very much, Luisa. We have a few questions. One that came from Faraz. It's about your GAP assay. The question is G12. So KRAS G12D is famous for being defective in its GTPase activity and refractory to GAPs. So how do you explain the reduction in GTP-bound KRAS G12D in your pull-down assays?
Great, thank you for the question. So we have known that KRAS mutants in general compared to the wild type mutants are less effective at being engaged by the GAPs. However, between the mutants, there is still a range of GAP susceptibility, and being G12D, the most susceptible by GAPs. This is work from other groups, from the DARE group, I'm blanking on his name at UT Southwestern. Anyway, so we know that the mutants to a certain extent are susceptible to the GAPs. What we think where we're seeing such a big difference is because there are so many GAPs being altered at the same time that potentially there is more feedback loops that are just not explained alone by one GAP binding to KRAS, but there's potentially other feedback loops that are regulating the expression or the active form of KRAS and other GPTases.
Okay, so there's another a second part to Faraz's question, which is in the in vitro assays, wild type and mutant KRAS have almost identical basal GTPase activity, which is surprising given that G12D should have several orders of magnitude less intrinsic GTPase activity. So can you comment on that?
Right, so this was normalized by, so we're talking about here, right? The wild type and the mutant. So for at least, I forgot to put it here, we added the other control, which is a known gap of KRAS, which is P120. The P120 has very different activities for wild type versus KRAS. So the P120 is very efficient at bringing down the wild type KRAS, while in the G12D, it's actually pretty much as not having any gaps. Well, GAP17 is not a canonical KRAS gap, it's a row gap. However, we're seeing that there's some form of activity and it's very similar between them. That's something that, we were discussing back with our KRAS experts, and they said that there's still a lot to understand about how there is potentially some spillover of row gaps to go for KRAS, as we're seeing here. But the biggest differences between wild type and mutant KRAS by a gap are seen with the control, which was P120.
Okay. Maybe we have time for a couple more questions.
Sure.
Tess asks whether the poly-C motif is specific to hnRNPK and whether there could be competition with other RNA binding proteins for the regulation of these exons.
Yeah, so as I was mentioning earlier on, and I think, Matthew, you were asking me about this, hnRNPK only explains a small percentage of these poly-C exon gaining. there is so much to understand about this policy exon gaining. We were saying we're exploring right now p53 directly interacting and masking or blocking these sites of recognition and at the end promoting policy exon, but we don't know. There could be competition with other RNA binding proteins, either neighboring, as we all know, RNA binding proteins don't necessarily need to bind to the motif that they're regulating. They can be downstream or upstream. So all of that beautiful biochemistry and RNA biology we didn't have. We didn't have the time, but that's something that we're interested in. We're following up on that.
Okay. And maybe one last question from Matthew, who asks whether you have evidence that mutant p53 alters hnRNPK expression in other cancers, or whether it's specific to pancreatic cancer. And you just mentioned, I think, 2 other types of cancer where this could be happening.
Yes, we didn't go back to other cancer types to see the correlation between mutant p53s and hnRNPK expression, except for PDAC. Where we did see the differences in KRAS activity by mutant p53, it's in other cancer types. So definitely that's something that we can do. We can go back to look at hnRNPK. And again, even if we don't find that tight relationship with hnRNPK, it doesn't mean that it cannot be promoted by another pathway, because we feel that we have not yet uncovered fully how these broad transcriptome-wide retention of policy exons is being induced by mutant p53.
Great. Yeah. Thank you, Luisa. Again, I think it was a beautiful story. There was another, okay, maybe one last question. We have 2 minutes. Coming from Osvaldo, he asks whether you have further explored the role of polypurine motifs, the poly-C motifs that you found alongside the poly-C exons.
Great. Well, that's a very good question. So basically what he's referring is, what are we going to do about this other side of the story that we haven't explored? We're actually in the middle of doing or figuring out what is now the consequence of these repressed exons, because right now we have only somewhat understood this policy exon retention, but not this flip side. So yes, so we are definitely doing this right now.